What were the biggest scientific differences between kind of using a patient's own cells versus donor-derived cells?
They have very different approaches in the clinic. The red cells are a non-nucleus cell type. They have a very short lifespan, very different than CAR T, where we wanted it to hang out in the patient's body. The red cells we used as almost a scaffold. We could put multiple different proteins, and because it's now membrane-bound versus soluble proteins can have different effects in the immune system.
What problem in cancer treatment are you trying to solve?
As an oncologist, I feel pretty strongly that chemotherapy and cytotoxic therapy, these toxic therapies are never going to go away. Of these more toxic therapies is we want to kill the cancer before we kill the patient.
Well, good morning, Chris. Welcome to the show. How are you today in a beautiful Philadelphia? I hope it's thawed out a little bit.
It is a beautiful day here in Philadelphia, and we're finally losing a lot of the snow that we've had.
Well, thanks for sending it this way. I'm in Boulder, Colorado, and we've had a very mild winter, and it's snowing today, and so I'm also in a sweater, but happy to see the moisture. But thanks for being on the show. I was really excited to be meeting with you. I know you've had such an illustrious career, and I thought I really wanted to dedicate some time diving a bit into your background. And I know that you trained under Dr. Carl June at the University of Penn, your early days of sort of the CAR T cell therapy days. And I thought maybe we could just start there.
I'll be honest with you, and Carl knows, and I tell this story and he agrees to it. At the time, there were really two sides of the June lab. There was the CAR T, which it wasn't even called CAR T. It was called the redirected cell therapy side. And then there was the active vaccine, where we were using cells to develop vaccines. And Carl, in some of my first interviews with him, because I was one of his first 10 postdocs, he came while I was a first-year oncology fellow. It was one of the serendipities of my career. He was describing how he was making these redirected T cells and felt like T cells could kill cancer. An immunologist and, PhD in immunology. So I was looking at that, and I honestly said to him, I said, well, I'm not sure that's ever going to work, so I'll do the vaccine side. So all of my, people look and they look at all of my papers from my postdoc, and they're like, there's nothing in there about CAR T cells. I'm like, yeah, I was the original one that told him I didn't think it was going to work. But it was, I'm telling you, it was a great place to grow up. 10 postdocs, 2 graduate students, 2 research associate, and we had a journal club every week. Like one of those things that you think to yourself, oh, if I could go back to just one journal club. Because Carl used to rip us to shreds, but it was great training, for being on a podium at a science meeting. It was a great place to grow up scientifically.
No, that's so impressive. And Chris, I think you're being hard on yourself, because I think the field at that time was also very skeptical about whether this technology.
I think it was, yeah.
Right. And we'll talk a little bit about that, because we're still seeing some challenges. But I think for listeners who might not fully understand, the various modalities and treatments, do you want to just give a brief overview of sort of, CAR T and sort of immunotherapy approaches and how they're different from traditional chemotherapies or targeted therapies?
Absolutely. I think that a lot of immunologists and immunology, in terms of its ability as a modality to treat cancer, it's gone through, this ebb and flow. The whole idea that cancer is actually a failure of the immune system is still a little bit controversial out there, and that one of the reasons that cancer cells continue to grow is that the immune system has lost the ability to recognize them, and that one of the systems that keeps us safe from cancer or eliminates very early cancer cells that develop is the immune system. I'm a believer that, in some diseases, that the immune system really can have a very strong effect. Now, is it going to work on its own? I don't think so. And even Carl will tell you that, that preparatory chemo that we give before CAR T are administered to patients, that it's actually critical to put the tumor down a bit and give the immune system a bit of a leg up. Now, when we think about CAR T and some of these advanced therapies, there's probably, at least with just CAR T, there's 3 different ways that we think about it. The first is, autologous CAR T, which means auto, meaning it's from the patient's own cells. And so go and have the pheresis, the blood comes out, we spin it, take out the T cells and put it back in on the other side. Those T cells are then, for that individual patient, are managed in the lab. We insert the gene that allows them to attack the cancer. Then in the middle here, we think about allogeneic CAR T. Now, that one is really tough because when you put a foreign organ, which a T cell, it's part of the organ of the bone marrow, but when you put a foreign cell in, the patient's own immune system is going to go after it very quickly. So we've had to, it's been a bit of immunologic gymnastics, I call it, just trying to get to the point that foreign cell isn't recognized. The newest approach, and Carl was right out there in the front with this, is in vivo CAR T, meaning now we can give the genes, we give viruses that will go in. So you use a virus to actually cure cancer. You give a virus to go in and infect the patient's own T cells and redirect the T cells inside the body, which is probably the next generation of these. But I always go back to that which I learned as a postdoc. And Carl, I still believe it has to be combined with some sort of truly cytotoxic therapy. The immune system with the cytotoxic therapy, we've seen it over and over again. The PD-1 along with chemo. Chemo is probably never going away. And it's a really good combination agent. Yeah, so looking at the CAR T field, all different ways of doing it. And we're trying to make the whole thing work out in the end.
So you've talked about a lot of different modalities in that response, which was a great primer for everybody out there. The whole concept of, for example, pursued and led development for autologous CAR T-cell directed therapies, and then at Rubius on allogeneic red cell therapy, right? So tell us a little bit about what were the biggest scientific differences between kind of using a patient's own cells versus donor-derived cells.
So they have very different approaches in the clinic. The red cells are a non-nucleus cell type. So a red blood cell doesn't have a nucleus. They have a very short lifespan, very different than CAR T, where we wanted it to hang out in the patient's body kind of forever, was the hope. The red cells we used as almost a scaffold, and we could put multiple different proteins. And because it's now membrane bound versus soluble proteins can have different effects in the immune system. And so the whole idea behind the red cells was it's almost like a biologic therapy. We're going to use it as a scaffold. We can put multiple proteins and activate, again, the immune system against cancer. It was a very interesting, it was a great place to learn. We had challenges in manufacturing. We had challenges because we were trying to do it during the pandemic. Probably the two biggest things that we learned while we were there is number one, a non-nucleated cell. The advance that Rubius made was that cells could now be administered coming through the pharmacy instead of a cell therapy. And that was really the key, because the clinical sites that didn't have ICUs and weren't taking care of COVID patients also didn't have a cell therapy lab. And so there's different ways, and I think we're now loosening some of those rules on these advanced therapies, and pharmacists are just as well trained as anybody that works in a cell therapy lab. And so I think that was a pretty big advance for Rubius. The other thing, it's always all about manufacturing and how to get these cells before they extrude their nuclei. It was sort of how to really kick them and get the cells to divide. One of the biggest challenges, whether it's autologous, allogeneic, is really finding ways to get those cells to divide so that you can get the dose that you want of the cells before it actually gets packaged and labeled and vialed up for the patient. So we learned a lot about cell therapy by implementing the red cells.
And it was interesting because I know one of the areas where we've seen success has been sort of in the blood cancer space, and then applying that to solid tumors has been so challenging. But at sort of one of your more recent companies at Immunicore, you did lead the development of KIMTRAC, which was actually a success story, right? Tell us a little bit about that, and what did it take to kind of move that complex immunotherapy and into making it commercially successful?
So it had a very similar feel to this in vivo CAR T in that Kimtrack and all the MTACs at Immunicore, they almost have two ends. It's a bispecific. One end is an antibody that is going to attack and find a cancer antigen. And the other end is designed to grab a T cell. It's almost just a linker protein that sits in a tumor. And then as any T cell flows by, it could be a flu T cell, any T cell that is directed at something, it gets directed then at the cancer. And one of the observations with the MTACs, and specifically with Kimtrack, which I always found so fascinating, patients would have massive responses with infections. Like you would have, just a bad cold. And all of a sudden, the tumors start to hurt. And it's because the T cells got activated. You can go from, 0.1% to 40% flu active T cells when one gets the flu. It's that plasticity of the immune system that I always found so interesting. Towards the end of my time there, I used to actually hope that patients would get just a cold, small viral infection, get those T cells going as they were being dosed. It was nothing that ever really, but it was just one of those anecdotal things that we just kept. So the immune system is so plastic. And I think there's so many different ways that we can use it, that we can leverage the immune system against cancer, which, I've always felt that the immune system is going to be part of the cure, ultimately, for cancer.
So, Chris, that mechanism you described with Kimtrack at Immunicore, that is that's kind of like, kind of what fostered your interest in joining Avacta at Therapeutics and using the technology you have there. It sounds like, you're going after this antibody drug conjugate space. Can you tell us a little bit about, what drew you to Avacta, at this point in your career? There's
a couple of different Immunicore links. The chairman of Avacta had been the previous CEO at Immunicore. And so he hired me, he plucked me out of Novartis in New Jersey over to London. That was my first gig in living in Philly, but working in London. So he hired me to the Avacta board and just said, look, we could use some help on the R&D side of things. As I was a director of Avacta, I can still remember the day and I can still remember the slide. It was when the first biopsy data came in, and we were sitting around as directors looking at the first time that we had shown that the Avacta peptide drug conjugate could concentrate a payload in a tumor at nearly 100 to 1 versus what we see in the plasma. And at the time, we didn't have any responses. We were actually probably treating the wrong tumor types in the trial. And I still remember looking at that data and thinking, wow, this mechanism really works. I went back and grabbed the board slides, so still have them from that day. I started then an interesting campaign with the other directors. I was like, I think they need someone like me. And I was somewhat unapologetic about it. I really wanted a job at Avacta. I really just loved the technology. It took some time, but at the beginning of 2024, I took over as head of R&D. And then by the middle of 24, the board felt like it might be time for a change in CEO. We were sort of pivoting away from diagnostics with those exciting data and moving more towards an oncology therapeutics company. Yeah.
Well, it's a beautiful story, and that also explains the strategic pivot that the company made and installed you as the leader there. That's really interesting. How would you describe to someone hearing about Avacta Therapeutics for the first time, what problem in cancer treatment are you trying to solve?
So, as I mentioned, as an oncologist, I feel pretty strongly that chemotherapy and cytotoxic therapy, these toxic therapies are never going to go away. But I think that there's much better ways that we can give them. The whole idea behind some of these more toxic therapies is we want to kill the cancer before we kill the patient. It's really horrendous, some of them out there. I was a pediatric oncologist, so I took care of everyone from 20 somethings down to babies. cytotoxic therapy is very effective. There's also plenty of targeted therapies that are very effective, but they're designed to kill, dividing cells, and they're designed to attack things about that biology of a rapidly dividing cell. Now, there are areas in the human physiology that are rapidly dividing: hair follicles, bone marrow, GI tract, and I just named many of the tissues where we see the toxic effects of these therapies. And so the whole idea behind a whole new, let's call it XDC, or any kind of drug conjugate, is to protect normal tissues from that drug. Mm-hmm. Take it a step further with Avacta. The whole idea is to concentrate the payload, the released payload, in the tumor itself. And that's what's slightly different about the Avacta approach, is not only do we want to shield normal tissues from this toxic effect, we actually want to concentrate it and drop it right where it should be. And so what's different about our peptide drug conjugate versus, the cleavable ADCs, let's call them, is we have a tumor-specific release mechanism. So Avacta leverages an enzyme that is actually expressed in the stroma of a tumor, which are the supporting cells that help keep and help wear off and protect the tumor from the outside influences, but they're called fibroblasts. And the fibroblasts express this enzyme on the extracellular surface. And so the peptide drug conjugate, it's a peptide attached then to the payload. And the peptide now has, I usually use my phone. And so if my phone is a molecule of an anti-cancer agent, this molecule doesn't know the difference between the GI tract, the bone marrow, and the tumor. And so it enters all of them easily and equally. We have a small little peptide, 2 amino acids, so it's unrecognizable by the immune system. And when we attach it to the drug, now you've changed the physicochemical properties of this. And now it can't get into the GI tract, the bone marrow, or the tumor. But that enzyme that I mentioned, FAP, in the tumor removes it. So you concentrate active drug in the tumor and protect normal tissues. And just that simple peptide leads to 100-to-1 concentration in the tumor microenvironment. And so at this point, we've been doing just oodles of chemistry around this, the peptide, and optimizing both sides of it so that we call it FAP enable, or attach the peptide to almost any medicine out there. And so seeing that, a few years ago in the board meeting, like, oh, wait, this works. Now what we have to do, we've got this brilliant chemistry team, but now we have to do is figure out how do we situate it in the FAP and still get the FAP to release and leave. And our chemists over the last, I would say, year or so, we've made a number of inventions and improvements. And so we're really at an inflection point now. The company, we've got our second drug is entering the clinic. It's a topoisomerase 1 FAP enabled. And if that one works like it does in the animals in the preclinical setting, I've been quoted as sort of the sky is the limit. And it's not just cytotoxics anymore. It's targeted therapies, that also cause havoc in some patients. And so I'm pretty excited. I've gone all the way from cell therapy back to small molecules. So it was a little bit of a pivot, but it's sort of what everybody's been looking for, which is how do we protect normal tissues and really drop that drug exactly where we want it to be in the tumor. As you can tell, I'm pretty passionate about this company and this technology, and we're excited. This next one really is going to show sort of the new way that we can use it.
Well, that was a great explanation, and really understand. I like your example with the cell phone and the mouse, kind of, and the peptide. And
I've used that a few times.
Yeah. I mean, it helps kind of visualize, what's going on. So let's talk a little bit more about the clinical pipeline. You mentioned, 2 assets. AVA 6000 sounds like your lead candidate, and you're going after doxorubicin, right? Which, for our listeners out there, maybe describe a little bit how doxorubicin is currently used and what its limitations are, and how AVA 6000, what you know about that drug and how it might be used.
So AVA 6000 is, we call it the stripped down version. So it's literally drug and peptide, no bells or whistles. So we know exactly how the peptide is going to behave in the clinic. Now, challenges with doxorubicin, It has a very limited activity spectrum. There's a very limited number of tumors that it actually works in. It's also not a terribly potent drug, but one of the advantages is that it's got a pretty long half-life of 35 hours. So at the time that we put that one in the clinic, we didn't believe that we were going to be able to impact half-life at all. So we needed something that could hang around for quite some time. What I mentioned is exactly what we see in the clinic. It all cleaves very quickly. The half-life of AVA6000, the peptide drug conjugate, is only 45 minutes. I'm going to go nerd on you for a second, but there's a measure of a substrate and an enzyme, and it's biochemistry, but it's called KCAT over K. It's just how efficient is this particular substrate for this enzyme? And that is easily adjusted in human biology. Well, AVA6000 is a very efficient substrate for the FAP enzyme. It all cleaves quickly. And because it all cleaves quickly, we do see then the released payload leaking into the bloodstream as we get to those higher doses. Now we're able to dose 4 times the dose of doxorubicin that is tolerated by patients in the clinic. The other thing that was important about the first drug that we took in is we wanted a toxicity. We wanted a key toxicity and not like neutropenia. We wanted a key toxicity that we could either eliminate or severely reduce. Doxorubicin causes heart failure, and it's a cumulative toxicity. So we usually stop when we use it, doxorubicin as monotherapy, we stop at like 6 doses because that's where you really start to see the risk. Now we've essentially gone well beyond that 6 doses with our drug, and we took those data to health authority, and we've now eliminated the lifetime maximum because guess how much cardiac toxicity we see? 0, nothing. Our pharmacokineticist gives a great explanation of why it's just not reaching the heart, but that was a pivotal moment for the company in that we really do have this separation and really worked on that therapeutic index. What is the concentration of toxicity and the concentration of efficacy? And the fact that we could eliminate that toxicity was critically important and something that we really needed to learn. So biopsy data, safety data, cardiac toxicity, cardiac exposure, all of these led into the next invention, which was our ability to turn that topo-1 exatecan, which is the most potent topo-1 that's ever been used in patients, but turn that one into a peptide drug conjugate. So that's the second one.
Gotcha. Just to follow up on AVA6000. So given kind of, where doxorubicin had been used today in breast cancers and things like that, tell us a little bit, is that currently where you're studying the product, and expand trials in breast cancer? And
so ironically, the first tumor type, which is very Kimmtrak-like uveal melanoma, but the first tumor type where we really saw just exquisite activity is salivary gland cancer. It's a subset of head and neck cancers. And it's also a place, very Kimmtrak-like, I will say, Chris, but it's also a place where there's just incredible unmet need. There's nothing for these patients. It's terribly chemo-refractory, so even chemotherapy alone doesn't work. And so it 90% disease control rate in patients where, you don't have a response rate with chemotherapy. So that one we're particularly excited about. And then there are a couple of additional tumor types we're thinking about. Soft tissue sarcoma is one, and we've put out some data there. Breast cancer, triple negative breast cancer, but there's also ovarian. So those are sort of the 4 tumor types. One of the lessons that I think the ADC field has learned as well, but for us it was payload sensitivity. And so we did treat patients with pancreatic cancer. FAP is expressed in 90% of solid tumors, but we didn't see any activity there. Well, doxorubicin doesn't work. And so that is a really critically important point for that particular drug and for this entire platform. Payload sensitivity is really going to be important as we move through and create new precision medicines.
Given the interesting results in this tumor type, the salivary gland cancer with high unmet need, what can you say publicly about kind of any kind of breakthrough designation status or whatnot from FDA? Have you received any sort of fast track designation?
So I would say stay tuned. And it's, we did put a little bit of information out in the public domain about the cardiac discussion. We did describe a few things there. We've also have a path forward to, really defining the dose. And so you can read between the lines. That would be a Project Optimus. So we've had a number of really good conversations, but yes, and we've got new data that we have committed to and before the end of the first half of this year. And those new data, especially in salivary gland, are so important for the company for a number of things that you just mentioned.
Yeah. Well, Chris, I realize, drug development's expensive, and I know that you've got another asset in the clinic. So, maybe how are you looking at prioritizing the trials and your assets going through development to sort of manage the cash burn and everything? Like, maybe tell us a little bit of how because I know we're going to talk about the other asset. I think that I'm not sure if you're bringing it into the clinic or if it's already in, but tell us a little bit about how you're managing the sequencing and to be efficient with your development.
Sure. So for AVA6000, we're still working through those expansion cohorts. We did just get that feedback specifically on Project Optimus. So that cohort is ongoing right now. That went up on clinicaltrials.gov. So need a little bit more time with that one. For a company, having done this before at Immunicore, transitioning from that first early trial into, the let's call it the larger suite of pivotal trials, the conversations are quite important with the health authorities. Those are absolutely ongoing. But the company would have to pivot and, hire and grow. And we've been very upfront that we're looking for a partner. We're having a number of very good conversations about this one. We think that the PFS, just looking at the figures from ESMO, the PFS is pretty exciting in this one and what we're seeing there. And so we are looking for that sort of level of help with AVA6000 so that, because we're really only 24 people there in White City and a few scattered in the States, and then pivoting to that next drug, which is we think, such a big inflection point for the company. So, love both of them, but yeah, the small clinical team, translational medicine team that we have, we really need them focusing on AVA6103, finishing up this stage of development with AVA6000 and seeking and moving towards a partnership for that one.
That makes perfect sense. So let's talk a little bit about AVA6103 that you mentioned. Mm-hmm. That one is getting ready to enter human clinical trials, right? It's not yet. You haven't yet started trials, have you?
No, but imminent.
Imminent.
Definitely
okay, so
imminent.
Tell us what makes that therapy exciting, and I would admit it's obviously a different payload. Tell us a little bit about what you're doing there.
So when we started to see the data with AVA6000 and how doxorubicin behaved, and how different it behaved, when you compare the Cmax of our released doxorubicin to traditional doxorubicin, they're miles apart from each other, and that really speaks to the toxicity. We also get that concentration. And so we knew that the platform worked. We also knew that if we were going to implement something, and exatecan was one of the drugs that I put down in front of Francis and his whole scientific team. Francis is our CSO. He's a brilliant chemist. We needed to develop a couple of things. The first is that we had to extend the half-life of the peptide drug conjugate. I mentioned 45 minutes to an hour. We had to extend that half-life, and I challenged them. I was like, get me 12 hours of this so that we get that. But the other thing we had to do was develop a sustained release mechanism. And that's where I gave them a real challenge, which was, this drug has a half-life of 9 hours. Can you turn that into 3 days? We kind of all had a little bit of a chuckle. And Francis, this is so inappropriate, but Francis, it was almost like, hold my beer, I'll be right back. It wasn't, but the chemistry team worked so hard. They took the peptide and they created two ends to it. One of them, they created a capping group, which sits on the outside, and it actually sits right in the enzyme. And then they created a linker. So the drug has to actually fit in the active site so that this gets removed by the enzyme and this falls out. And so we borrowed an idea, a technology from the ADC field, which is a self-immolative linker. Self-immolative meaning as soon as this piece of chemistry is exposed to the bloodstream, it just falls apart and the drug sails away. And so those two together took this drug from a 9-hour half-life when it was developed in the early thousands or the early 2000s, late 90s, early 2000s, and created a scene where now we see 5 days of release within the tumor. It was, and I've told this story before, so no, I don't think they'll get mad at it, but I've accused them over Zoom of being so British because they showed me the data one day and it was 5 days of coverage. And I was like, you guys are so British. And I was like screaming in my kitchen and so excited that we had seen this. And I just loved it. Our head of ClinOps at the time, she's Scottish. She's like, well, I'm not British, I'm Scottish. And I'm dancing in my kitchen too, Chris. So it's such a fantastic, small little team. And I've used the Margaret Mead quote about them, never doubt that a small group of thoughtful, committed citizens can change the world. Indeed, it's the only thing that ever has. Tiny little company, it's us versus, everybody. But it's a truly amazing technology. And that's why I mean that this drug entering the clinic right now is really, it's the inflection point for the company. Because if those two inventions, the capping group and the linker, those two inventions, if it actually does what we think it's going to do in patients, watch out, here we come.
Yeah. Well, I love that how your team exceeded your expectation on the 3-day target product profile where you said, give me 3 days, I gave you 5. So just that's impressive.
Yeah.
Where are you going with AVA 6103 then? What do where do you, when you enter the clinic, if you've disclosed it or can disclose where you're looking to start that first or second trial and what patient populations?
So we did an experiment. I'm a big data kind of girl, and I'm a believer that, there's lots of data out there. And if we can leverage what's out there, there's a large database called Lens that is with a company, a data company called Tempus. And what we did is we went to them, and there's a gene, schlafen-11, that predicts for sensitivity to a topo-1, any topo-1, this mechanism. And then we have another gene, FAP. And so we asked Tempus to go into their database of all solid tumors, 160,000 solid tumors. We had developed cut points based on immunohistochemistry. And we just asked that, can you apply this and can you rank order all of the solid tumors in terms of which ones had the most likely chance of responding? And so there was a whole smattering of diseases right up at the top. And so we chose 4. And there we chose based on, the Tempus analysis, based on prior activity of topo-1, FAP, and whatnot. And so the 4 diseases that are going in first are small cell lung cancer, gastric cancer, cervical cancer, and pancreatic cancer. If you think about it, there's 2 small diseases. Small diseases and a small company like ours, they get along really well. It's a very easy, straightforward path, and that's small cell and cervical cancer. And then 2 larger disease settings, pancreatic cancer and gastric cancer. I've been asked the question, which do you think is the most likely? Well, if you look at Enhertu in gastric cancer, Enhertu is one of the best-selling ADCs out there, wildly active, works really well in gastric cancer, and we have almost the exact same payload. Deruxtecan and exatecan, they're almost exactly the same drug, slight differences. Targeting HER2 and dumping deruxtecan works very well in gastric cancer. Gastric cancer HER2 positivity is only about 15 to 20%, whereas 100% of gastric cancers express FAP. So if I had to pick one, I would probably say that gastric cancer is our most likely. But we're super excited. Then there's 2 diseases that are coming next for that one. We show activity or get through the dose escalation in those 4 diseases. ER positive breast cancer, non-small cell lung cancer are 2 of the next ones on that the magic list that we developed with Tempus. So there's a lot. Topo-1 is just it's a very widely active mechanism in oncology, So there's a number of places that we can take this one. I always mention colorectal. It's a very heterogeneous disease, but I do think that there's probably a nice subset of colorectal that would work as well.
Well, those are all tumors types and with patients where there's still tremendous unmet need. So they all make complete sense. Maybe just to ask you, based on your current limited but clinical experience working with investigators and treating oncologists, what are they saying? What resonates with them most clinically, and maybe what skepticism do you still need to overcome with that community?
So AVA 6000, I always say doxorubicin, having been an oncologist, doxorubicin is nobody's favorite drug. Mm-hmm. Nobody. It's wildly toxic. It has that cardiac toxicity, which it really does wreak fear in some oncologists' mind, especially when you're using it as an adjuvant therapy, which is where breast cancer treatment is using this drug. I'm going to go nerd on you a second again, but that very high KCAT over KM, that very high efficiency of the enzyme, I think about it like the tumor, like a painter's sponge. And if you put on the faucet of water, it doesn't end up leaking through. And that's exactly what we see. There's doxorubicin that leaks into the bloodstream. And we needed to fix that in the second drug. The exatecan is much more potent than doxorubicin. And so with this new chemistry, we think, based on the animal models, that we're really going to be able to protect the normal tissues. But that's probably the biggest skepticism. Are you going to be able to make that pivot from, with doxorubicin, with your first one, we did get some exposure. We had to stop at 4 times the dose level because we had, exposure and safety concerns. How has the chemistry actually fixed that problem? And that's why this is truly the inflection point for the company, 6103. With those inventions, have we fixed what we needed to fix with that first version of it? So that is probably the skepticism, because it's a potent drug. Exatecan is a potent drug. At the MTD, 90% severe neutropenia, 90%. So it's a toxic drug, and I hand it to Daiichi. It was originally owned by Daiichi, and in the 1990s, they tried very hard. It was a very active drug. Mm-hmm. I mean, it shrunk tumors as a single agent. It even in a breast cancer phase 2, Chris, it shrunk breast cancer tumors that had already seen prior topo 1s. That's how potent this drug is. So we've got to protect the normal tissues and be able to get that concentration up in the tumor. If it works like it did in the animals, I think we might be seeing something interesting in the clinic coming up.
Well, that's exciting. I think maybe on that note, could you summarize over the next year, maybe 2 years, given kind of where you're at today, what milestones should we be watching out for from the company? So
we are, AVA 6000, new data coming out towards the end of the first half of this year. And those are the data that are going to really show us, is salivary gland cancer, is that the right first indication for that one? Excited to get those data out there. 6103, that new chemistry, the exatecan molecule, that one will get started in the clinic in the coming weeks, and looking for a signal for that one, not completely finishing the phase 1, but a signal later this year. And as I mentioned, quite potent, quite toxic. We will know pretty soon about the toxicity for that one. We'll also be getting biopsies, so we'll be able to tell, are we getting that same, I'm doing the figure for it, but that tumor to plasma ratio going to be important. The other thing that we developed out of all of that chemistry is Francis and his team were able to insert in that linker a second pod. And so now, one of the criticisms is, well, your drug to antibody, your DAR or DPR, drug to peptide ratio, is only 1-to-1. Well, now it's 2-to-1 because we got another pod in there, and that's probably sterically. But now we can hang off 2 different drugs, the same drug. We published that in October. And so now we can use something like a topo 1 and come in with the resistance mechanism. That one I'm wildly excited about. We'll be looking for an update on we call it the dual payload. It's the first dual payload peptide drug conjugate. The dual payload ADCs are in the clinic already, but we're moving that one as fast as we can, because personally, I think treat with the agent and hit the resistance mechanism at the same time. So we're super excited to get that one going. There'll be lots of updates through the year on the dual payload technology, and we hope to have that in the clinic. That could be hitting IND studies or IND enabling studies either later this year or early next. Which, if you look at it took us only about a year from 6103 going to IND enabling to get into the clinic. So. The team seems to, it's only 24 people, but boy, do they seem to have it down in terms of, and we learned so much with 6103 on the pipeline, on the platform, on manufacturing. These are small molecules, so they're fairly straightforward in terms of their manufacturing. So yeah, lots of updates coming out of the company coming up soon.
Oh, that's awesome. Really impressive at the speed, the velocity of what you're accomplishing with a small team. So I would say you mentioned, you're open to partnership discussions. So for those listening who may want to reach out to you, what's the best way for folks to get in touch with you, Chris?
So they can go to our website. We have a partnering page there, but my email is chris.coughlin@avacta.com. And we're more than happy. We've got a lot of really good conversations. And I'll tell you, some of the advances that we've made have been from questions from potential partners. Like the dual payload actually came from a BD meeting. It was a potential partner who said, "That's interesting, but can you put 2 on?" And Francis, again, hold my beer, be right back. I mean, our scientists, our clinicians, they're all they're brilliant. We just have such an amazing team at Avacta. Very committed group of people.
Well, it sounds like an amazing team, and I mean, you seem like you're an amazing leader. I'm sure it must be a fun place to work. I can sense the passion in your voice, Chris. It's been a pleasure hearing your backstory too today. Thank you for sharing all that background, because I think it was very relevant to the work you're doing today. And
thank you so much. Appreciate the opportunity. It was a great conversation.
Yeah, thank you. Bye-bye.
If you enjoyed this conversation, be sure to subscribe to the RX for Biotech podcast to hear directly from the leaders who are shaping the future of personalized medicines. And if you or someone you know is navigating a care journey, be sure to visit therapyforme.ai to search for a specialist near you. And if you need that extra support, be sure to schedule a virtual telehealth appointment with one of our nurse navigators who can help answer your questions. Thank you for listening.